Hello, my name is Jennifer. I am a master's student and I will present the article. Is the efficacy of biological control against plant disease likely to be more durable than that of chemical pesticide? The durability of a control method for plant protection is defined as the persistence of its efficacy in space and time. It depends on the selection pressure exerted by its own population of plant pathogen, and in second phase, on the capacity of this pathogen to adapt to the control method. Erosion of effectiveness of conventional plant protection methods has been widely studied in the past. In contrast, the durability of biological control has long been assumed to be higher than that of chemical control. However, recent results concerning pest management in agricultural systems have shown that this assumption may not always be justified. The most intriguing example may be the development of resistance to the most widely used bioinsecticide in the world, resistant of viral pests to one several toxin of Bacillus thuringiensis. The durability of biological control of plant disease has hardly been studied and no scientific reports provide the loss of efficiency of biological control agent against plant pathogen. Its practice has been published so far. Knowledge concerning the possible erosion of effectiveness of biological control is essential to ensure a durable efficacy of biological control against on-target plant pathogen. Thus, the objective of this article was analysis of the scientific literature was conducted and scientific papers reporting the diversity of efficacy of biocontrol agents toward plant pathogens or tools describing the ability of plant pathogens to produce natural mutants with reduced susceptibility under the selection pressure exerted by a biocontrol agents were analyzed. Possible loss of efficacy of a biocontrol agent could result from the pre-existence of isolate with low susceptibility in natural population of plant pathogen. So in the article, the authors show the works approach the variation of sensibility of plant pathogen. This table highlight example of the variation in vitro. One example that I want highlighting is the compound 2,4-DAPG by Pseudomonas fluorescens against Fusarium oxysporum. And the pathogen show the 70% of isolates naturally tolerance and 10 times more resistant. 10 is the resistant factor of the 70% of the isolates. In the table 3, examples highlight the variation in sensibility of plant pathogen with a single mode of action. I can highlight the antibiosis with Agrobacterium rhizogenes against Agrobacterium tumefaciens, and the biotype 3 are resistant to antibiotic agroxin 84. And about hyperparasitism, the Cyphonectria hypovirus 1 CAV against Cyphonectria parasitica not show difference in tolerance the plant pathogen and show the small magnitude of tolerance. In the table 5, show the effect of formulate, the plant extract from Fallopian sachalinesensis applied at different concentrations of the spore germination of 3 different spored millet species on solid millet. This is about combination several modes of action. And the phallopsia have a different mode of action, for example, induced resistant and inhibitory effect on spore germination. But the results show that sporulation by some isolate was observed on the leaf disc, suggesting that use of this plant extract on a commercial scale could possibly lead to the selection of resistant isolates. About capacity of plant pathogen to adapt to biological control, one example is Bacillus subtilis CL27 producing three different antifungi compounds, two of which were effective in vitro against Botrytis and following 8 successive treatments on plants of Astilbe hybrida, the protective effect of the biocontrol agent against the pathogen strongly decreased and the control become totally ineffective after 10th treatment. Resistant to compounds produced by microorganisms. Is three examples the article showed. Excretion, ABC, ATP building cassette, or MSCF, major facilitate or superfamily transporters. Metabolization, the examples is the increased production by the fungus of catalases, peroxidases, and others, allowing the degradation of the antibiotic and protection of the fungus, and biosynthesis inhibition, that these interfere with the biosynthesis of antimicrobial compounds can also be a means for plant pathogen to be resistant to biocontrol agents. About resistant to cell wall degrading enzyme produced by biocontrol agent, specific mechanism can help plant pathogen like the synthesis of melanin polymers, chlamydospores-like structures maintain a slow growth and the production of conidia in presence of Bacillus subtilis. The mycotoxin zone produced by Fusarium culminarum and Fusarium graminearum represses the expression of genes coded for chitinases and the biocontrol strain Trichoderma troviride 1. Resistant to defense compound produced by the plants. The article shows again excretion. The example is secreted then out of the cells, as is the case of antimicrobial compounds produced by biocontrol agent, for example, ABC transpound, the same, and the metabolization, Botrytis cinerea is, for instance, able to detoxify the alpha-tomatin, a phytoanticipin found in tomato plant, and for the last, protection against oxidative damage. And he showed the HR facilitates the colonization of the plant by necrotrophic fungal pathogen. And he showed two examples of reduction population of biocontrol agents. He speaks about pigeon species, the population of Pseudomonas fluorescens decline by reduction the root surface available for bacteria colonization, which has a consequence, tends to limit the population of a potential antagonist. Knowledge about the plant microbial like rhizosphere, phyllosphere, and endosphere is important, and the microbial interaction occurring on the those niches should contribute to the improvement of biological control. The conclusion in the article is plant pathogen can display various, include very low levels of sensibility to biocontrol agent, regardless of the complexity of their mode of action. Certain pathogens also have the potential to adapt in a few generations to the selection pressure exerted by a biocontrol agent. It is a first step to evaluate the distribution and the potential impact to resistance in the field. Moreover, the detection of partially resistant isolate may indicate a risk of developing a more severe resistance that subsequently may initiate a possible loss of control in the field. Cross-resistance with other existing biocontrol agents or possible fungicides may also be determined. The context of the integrated pest management is probably an effective way to reduce the risk of resistant development, but it necessitates to evaluate the compatibility of the biocontrol agent together with other protection methods. Thank you, and pathogens stay away.